Happy March. Welcome back to those of you who are returning, and welcome to those of you who are first time here to Brigger Hall's auditorium, where today we have Professor Andrea Graham from the Department of Ecology and Evolutionary Biology. Andrea is a evolutionary biologist, you'd say, yes, who received the PhD from Cornell, and after a few postdocs in Great Britain, right, Scotland mostly, yes. Came to Princeton, and currently spends, and for a while has been sort of using ideas in evolutionary biology to understand sort of host pathogen interactions in a number of in evolution, things like that. But in seeing the bio, I saw a really exciting thing that happened early in the career, which was working with koalas. And I think that whenever anyone thinks about what they're going to do as a biologist, there's that check, work with koalas, done. So, and then on the other side of what you want to do when you work as a biologist is be named a fellow of the American Association for Advancement of Science, right, which was done as well. So really, check, check. And so this presentation is the third check of the trifecta. Yes, so thank you very much.
Thank you. Yes, indeed, delighted to be here with you all. So I am indeed an evolutionary biologist. I might even say evolutionary ecologist who really loves to think about the cells and the molecules of the mammalian immune system, which are what we're looking at in this photo here on the left. I do tend to study them in lab conditions, controlled experimental conditions, such as this Petri dish, but ultimately I want to understand what they're doing in the bodies of wild animals. And so that's why I always aspire to understand immune function where the wild things are, with thanks to Morris Sendek for these charming wild things who look just as fascinated by those cells and molecules as I am. Today, given the audience and the time allotted, I thought what I would do is talk to you about two mammalian systems that we work on in my lab. We study laboratory mice that get to spend their first days outside. We call them rewilded mice. And at the other end of our spectrum of wildness and body size, we have northern elephant seals. So there's about 100,000-fold difference in body size between the two host types that I'm going to tell you about today. They, of course, have lots of other things that differ between these species, but what they have in common are some of the ways in which particular cell types of their immune systems respond to cues in the environment. Before I go any further, I have to acknowledge with a full heart that everything I do is hugely collaborative, so I'm up here representing a wide team of collaborators, including many wonderful folks, past and present, working in my lab, and I will call them out individually as I'm talking about aspects of their work, but really I've been very lucky since I came here to have fabulous lab members. We also have amazing collaborators working in fields like ecological theory, molecular and cellular immunology, parasitology. Down here we move into the neuroscientists, the veterinarians, and the behavioral ecologists. We really need this diversity and excellence of team for the work that we do. We've also been lucky to have quite a lot of funding support from a few different New Jersey state sources as well as federal sources. And within Princeton, we've been also very fortunate to have the support of multiple units, Princeton Catalysis Initiative, Center for Health and Wellbeing, and last but certainly not least, High Meadows Environmental Institute, who has supplied us with some of the most fantastic interns for our field seasons. So with thanks to this entire slide full of people and units. So the talk outline is going to be as follows. First, I'm going to give you a bit more of a sense of what I mean by where the wild things are immunologically. I'm going to give you a brief introduction to how I think about immune function or why I think it's so fascinating and delightful to study. And there will be a starring role for a cell type called neutrophils, cartooned by that little purple guy up there. You will see those appear throughout the talk. And part of what I'll emphasize here in the introduction is how the genes of the host, the genetic background of the host, and the environment in which that host is living shape immune function. Then I will move on to the two case studies. The rewilded mice is sort of a biomedical case study for undertaking investigation of how genes and environment interact to shape immune systems of mammals. And then at the end, I will move on to the wild northern elephant seals as a conservation case for understanding genetic and environmental contributions to immune function. And then I'll conclude with an outlook and open questions, including many, I hope, from you. Okay, so the immune system is this marvelous thing that does not have an analog of the brain that coordinates the system from top down. Instead, the distributed autonomous cells of the immune system, in their trillions, must self-organize, more of a bottom-up coordinated system, in response to environmental stimuli. So these cells, the trillions of cells of our immune system, each with receptors on its surface that allow it to perceive and respond to cues in the environment, they zoom around our bodies. And in addition to the hundreds of lymph nodes, the little green dots are each lymph nodes, the hundreds of lymph nodes around our body, and the miles of lymphatic vessels in our bodies, they also can traffic through the circulatory system. And they're tumbling along there with their receptors out, perceiving different cues that come into the body. So what are the neutrophils doing? I'll tell you, give you a sense of their journey through life. First of all, neutrophils represent about half, around 50% of the cells circulating in your blood right now. What they do is they tumble along in the blood vessels. So this is sort of a cross-sectional cartoon of the blood vessel, and here's a neutrophil tumbling along with some of the receptors on its surface. And they deploy into tissues when those receptors pick up signs of an infection or an injury or some other problem that needs their attention. They squeeze out between endothelial cells into the site of infection or injury. And one of the most amazing things they do for us when they get there is they devour microbes. If you've got an infection because your cat scratched you this morning, you've got neutrophils in there gobbling up the microbes that might otherwise cause you some trouble. And so this is a non-cartoon but colorized scanning electron micrograph of a neutrophil to give you a little bit more of a sense of how charismatic and important they are. This is a micrograph of a neutrophil that's colorized in yellow consuming anthrax bacteria, which are colored orange. And you can see what awkward things those bacilli are to eat. And nonetheless, this single neutrophil is able to ingest quite a few of those bacteria. And boy, if you have anthrax, you're glad your neutrophils are doing that sort of thing. They're really quite extraordinary at helping us to control microbial infections. Another big thing that they do is that they produce chemical signals that recruit others to the site of the infection. A key example of those chemical signals are cytokines. So an individual neutrophil deploys to the site of infection, starts scarfing up the bacteria, and secretes cytokines that recruit others to the site. And this responsive collective of cells in response to environmental cues is what really makes a system of the immune system. Again, there's no boss from on high. They're coordinating themselves with these messages. And this is a really interesting image taken by the Lammerman lab at Max Planck. Every green blob is a neutrophil as it's crawling. They have kind of amoeboid behavior crawling into the site of infection that they've imaged with, I think, 2-photon imaging here. And the rainbow trails are the paths that individual cells have taken as they come together in this swarm to collectively help defend this host. So this is what happens in an acute sort of infection event. As you might imagine, once they've gobbled up the microbes that have invaded, this gets resolved, the cells sort of return to other duties, and that's all good. We want that to happen. But there are some effects of this response to environmental stimuli that accumulate over time, and indeed can accumulate over the whole lifetime of a host. So just last year, led by the wonderful Jessica Metcalf, my colleague in EEB, we started trying to map out what the accumulated life experience of an individual human host might look like if you think about their immune phenotype. And so this is a visual representation of quite a lot of literature from this paper, where the y-axis is some aspects of immune phenotype. So the cell type distribution, what those cells can do, how quickly then they can phagocytose, whatever aspect of immune function you would like to think about on that y-axis. And then the x-axis is the human life course from conception through to senescence. And what the purplish blob is capturing is that there's quite a lot of variance in immune phenotype that is evident sort of in utero. Very soon after birth, there's what people term immune education, where exposure to the microbes of the world, for example, as well as starting to eat food, starting to get vaccinated, all that stuff, causes a bit of a convergence after the wide variation of immune phenotype observed in the very, very young. But then over the rest of the human lifespan, childhood, adulthood, through to senescence, this then expands again, because of course individuals are accruing very different sets of environmental exposures and experiences over that time. And the reason the neutrophil gets another appearance up here in the top right is that they actually have some very important roles to play in late life health that are not so great, frankly. There's a process that people have termed inflammaging, where there's sort of chronic high-level inflammation, and the neutrophils are participating in that as well. So we love them, but they do cause us a little bit of difficulty. Okay, so environmental experience is really driving the development and as well as the system-level variation in immune responsiveness among individual hosts. So given all that, it won't surprise you to hear that no matter what wild things you want to study, whether those might be human wild things or mice or elephant seals, they exhibit really varied immune defenses, especially once they've hit adulthood and beyond. There are genetic contributions to this, for sure, but environment is also a very important one, as well as other factors such as age, sex, and what's called modulators here, sort of chemical modulators. So sometimes immunologists who work in quite reductionist experimental settings look at this and tear out their hair, because there are so many causes of heterogeneity here to try to wrap your head around. But evolutionary ecologists like me get very excited, partly because we like to try to dissect the proximate explanations for variation in immune phenotype, the relative and interacting contributions of these factors to the varied immune responses you will see. But also, actually more to the point, we get very excited because these can present the raw materials for natural selection, for understanding how natural selection will operate on immune systems, especially when they're interactive effects, i.e. it's not that the genetics matter, it's not that the environment matters, it's not G or E, but some combination of the two, interactions between things like genotype and environment. So for example, in the case of the neutrophils, genetically encoded variation in the receptors that the cells are expressing may determine different responses to particular environmental stimuli that those receptors encounter. And more on that as I get into the examples, the case studies. Okay, so off we go into case studies. So the first one is our biomedically motivated case study. So I've just told you that genetic variation and environmental variation and more shape immune function in the real world, but this is the system that is the source of just about everything we think we know about mammalian immunity, because experiments in biomedical immunology use inbred laboratory mice kept in extremely, most of them, kept in extremely dull, constant environments with things like irradiated food and autoclaved bedding. And this is actually disposable caging so that it can be, there's no signals from the cage that are passed on. This is very much where the wild things are not. And I have an exclamation point over here because despite how divergent this experimental setting is from the real world where immune systems function, we've learned a lot about mammalian immunity from those laboratory mice. Things that generalize to other species, things that generate treatments in human clinics, right? So that it gets an exclamation point because it's quite amazing how much we have learned. But it also gets a question mark because I have long wondered whether we can do better and do more and learn more from the laboratory mouse. In particular, I'm really curious whether we can improve experiments using inbred mice, but give them life course experience, allow them to accumulate interesting environmental experiences. And neutrophils make an appearance here again because people who like to criticize the use of laboratory mice in biomedical research often say, well, the number one thing is they don't have enough neutrophils. I told you that we all have around 50%. It's as low as 20% in the laboratory mice that live in these super clean cages. And this just doesn't, frankly, make them seem like respectable adult mammals on Earth, frankly. So I and my collaborators wondered whether we can fix some of those things by putting the mice in varied environments. We've continued to work on inbred mice, so taking advantage of the tools available for the various genetic lines of laboratory mice that have been developed for biomedical immunology, but we put them in varied environments. In fact, we put them in a natural environment that varies in many factors at the same time. They're out there in the New Jersey heat and humidity in the summer. They get to chase crickets for the first time, and one of our collaborators is studying how they very rapidly become good hunters. They also get to burrow in the soil and are exposed to microbes in the soil. Okay, so this means that in our summers since 2014, we have been addressing this question: how do experiences accumulated outdoors affect the immune phenotypes as well as disease susceptibility of these inbred mice? In terms of human resources, the person who, from day one, has been helping me to make this happen is Billy Cragans, who manages Stony Ford, which is EEB's research station. So he has been absolutely essential to all of this work, so thanks to him. But we also have some hardware that has been essential. And the number one thing, this is the Google Earth view of our mouse enclosure facility, or our mousocosms. We have 8 replicate pens into which we can release mice. This whole sort of octagon-shaped thing is a little bit bigger than this room, to give you a sense. We've mostly, let me get to the other important piece of hardware first. So this is a Longworth mouse trap, a live trap. As you might imagine, unlike biomedical scientists working with mice in the cages who just open the lid and pop out a mouse when they want to sample its blood or something, we can't do that when the mice are in this very large facility. And so we use classic trapping methods from wild rodent studies. And that's our Longworth trap hardware, without which we couldn't do this work. So we have mostly studied young adult females, and we've kept them outdoors for up to 8 weeks in our experiments so far. We would love to go beyond that, but that's where we've begun. To give you a slightly closer up view, now down inside one of those wedge-shaped pens, you can see that the mice, we give them a little hut. We let the meadow plants of Stony Ford thrive. We have these tall steel walls that also go underground. We have electric fence on the perimeter, and we have bits of aluminum foil on fishing lines strung up in the air, which all sounds very elaborate, but in short, we're trying not to study predation. And of course, mice are on everybody's menu out in the real world. And we have great success, actually, in recovering the mice to study their immune systems. We also quantify quite a few aspects of the environment that they're experiencing, including abiotic factors like the heat and humidity, but also very much including the microbes that they get to experience for the first time when they go outside. So to give you a sense of the data that we're able to collect out there, first of all, on the topic of those microbes, the symbionts that live in and on the mice, we find both in their guts and on their skin, thanks to Yoon. A graduate student in my lab brought that angle into the lab. Gut and skin microbial diversity is rapidly increased when the mice go outside, and then it persists at a higher diversity. We've done so far both bacterial studies and fungal studies, and fungi show up in red font over there because fungi are about to have a starring role in what I'm about to tell you. But we also know that in addition to this increased bacterial and fungal diversity that they're awash in, they are consistently over 30 unknown mouse pathogens. We do keep them away from wild rodents too, so we're not surprised by this, but it's sort of interesting to note that everything I'm about to show you are animals that, yes, they have a much richer life of exposure to environmental microbes, but they aren't catching infectious diseases per se. Okay, so we've done quite a bit of work on this over the last several years, done lots of different aspects of immune phenotyping, T cells, B cells. If I'm not talking about your favorite cell type, please ask afterwards, but I'm going to continue my focus on neutrophils. This is work that's been led by Yinghan Chen, who at the time was a postdoc in the lab of Ken Cadwell at Penn, but who now runs his own lab. He's now a professor at the University of Taiwan. He did the work to show that neutrophils really do get boosted by time outdoors. Suddenly a mouse, after just a month or two of exposure to the real world, has neutrophil counts that make them look like those respectable adult mammals we knew they could be. And he went on to show, with a lot of very impressive culture work as well as flow cytometry work, he showed that fungal exposure to soil-dwelling Aspergillus and Talaromyces, growing in those colony forms shown there, really boosts bone marrow production of neutrophils. Something about those exposures just ramps up the entire neutrophil production apparatus in the femurs of the animals. So we're here comparing lab versus rewilded. These are various myeloid progenitor cell lines that are in the bone marrow, and MPP3 is well known to be what powers neutrophil production, and sure enough, in the bones of these mice, their neutrophil production has really ramped up. They also have those high levels of neutrophils circulating in the blood. We've also studied all those other aspects of immune phenotype in diverse strains. They're all inbred, but they are selected and inbred to represent a lot of standing diversity in the Mus musculus species. This work was led by Bola Oyashola when she was a postdoc at the National Institutes of Health, in the laboratory of Peng Lok. She's now on the faculty of Penn Vet School. In collaboration with my former graduate student, Alec Downey, who is now a postdoc over at a Max Planck Institute in Germany. Those two visionaries led the experiments on these more diverse mouse strains, and basically they picked, these are the collaborative cross-founder strains that are bred up by the Jackson Labs for immunology research, basically picked 3 of the pigmented ones and released them outside and compared how life outdoors affected the immune phenotypes of these different strains. And again, excitingly for an evolutionary ecologist, the effect of the environment on the immune system depended upon which genotype you were studying. So the whole genetics by environment interaction that evolutionary biologists love to think about. We're in the process, yes, that shows that all the complexity of the wild thing immune variation that I showed you is happening right out there at Stony Ford with our strains of mice. We are in the process of unpicking exactly what the differences among those mouse strains are in terms of the receptors that their cells are bearing, for example. But we are also really keen to begin to predict the trajectories of effect of environment on the mice. And this work is led by David Chang van Oort, a current wonderful postdoc in my lab, in collaboration with Clay Kressler at the University of Nebraska and Dan Metz at Franklin and Marshall. And what we're trying to do is use mathematical ecology to model the development of an immune response and to understand how time outdoors powers these very divergent effects on immune phenotype. And in particular, those cytokines, the signaling molecules that the neutrophils produce, we're studying their cytokine feedback loops, the ways in which the signaling molecules among the different cell types synergize to generate the system-level response. And we're trying to understand if that allows us to predict the trajectories of those environmental-induced immune variations. So love to talk more about that if folks are interested. But one snapshot is that we do find some power to predict the immunological future as follows. If we measure immune cell distribution on the day we might give the immune system something to do, like when we challenge the mice with this gastrointestinal parasite, after they've been outside for a couple of weeks, we trap them and then we give them a worm to work with. If we measure what their immune systems had done in response to the outdoor environment that day, we can predict their worm burden 3 weeks later. As you can see, it's kind of a mixed bag depending on dose, depending on cell type, but just highlighting the neutrophils right up here, it turns out when the mice are given quite a low dose of parasite eggs, high neutrophil counts on the day those eggs arrive predict higher worm burden than other mice might get. So we're very excited about using the combination of mathematical tools and our data to understand how those trajectories of immune development and what their consequences are for disease progression. So I have a synopsis, and then I'll dive into the elephant seals. So Fungus is Among Us is another children's book that captures the spirit of this talk very well, I think. In short, the fungi in particular that mice encounter outdoors induce lots of neutrophil production. It happens in some host genotypes more than others, so that's that genotype by environment interaction. And we also find that how those neutrophils are boosted by time outdoors predicts the immunological future. It predicts susceptibility to subsequent infection. The fungus is among us. Okay, so now I'm going to shift gears to talk about our larger hosts, northern elephant seals, who just get an exclamation point for their very being, right? They're incredible. But they also get a question mark because their neutrophils are also a little bit of a mystery, as you will see in the next couple of minutes. So northern elephant seals were declared extinct in the late 1800s because they were overhunted. When the hunters ran out of whales to shoot, they started shooting northern elephant seals. But about 100 of them were found off the coast of Mexico in 1892. And so they had not gone extinct, and now there are estimated to be about 200,000 of them that have bred up from that population of about 100. So this might look like a terrible scene, but this is them just catching sun. But I do have to confess, I don't know how many of you heard this, just in the last few days, this very colony has tested positive for H5N1 bird flu. We've been worried about this for a long time because, first of all, the seals are susceptible. They share their habitat with lots and lots of birds, but in particular, the southern elephant seal, a closely related species, was really pretty decimated a few years ago by bird flu, and I'm sad to say that the northern elephant seals just in the last 5 days have tested positive. Most of them, they had just finished their breeding season, and so very few of them are actually up on the beach right now, and we're hoping that will help them to withstand the avian flu epidemic. More on that coming soon. Okay, so back to the story that I was intending to tell before avian flu appeared. So they came from a bottleneck of about 100 individuals. They're now up in their hundreds of thousands. This all sounds really wonderful, but the folks at the Marine Mammal Center in Sausalito, who specialize on looking after stranded marine mammals and bringing them back to health if they can, started noticing something very disturbing. And I have a little warning message down there in the bottom because this next slide is pretty gross. They started noticing that northern elephant seals who were stranding, especially quite young ones, pups and yearlings, were dying of a lungworm infection. And it seemed to be when they, at postmortem, they would study the lungs. These are the lungs of an elephant seal who has passed away due to lungworm infection. They basically saw these huge lesions all over the lungs associated with really high neutrophil counts. Stephen Gorin has been leading some bulk transcriptomics of this lung tissue at postmortem, and that confirms not only that the neutrophils are there, but all the cytokines that neutrophils can make and cause mayhem are there too. So the vets called us here because they knew that lung inflammation and what they called disseminated coagulopathy, sort of clotting of the blood all across the body, not worms per se, were killing these seals. And this was a surprise because other seal species get lungworms and they're okay. This is the lungworm life cycle. A seal gets infected when it eats an infected fish. After the parasite migrates around, ends up back in the gut, it produces larvae that the seal can excrete. A fish can eat those larvae. The nematode larvae develop further in the fish, and then the life cycle is completed when the next seal eats the infected fish. So harbor seals, for example, are part of this life cycle too, and they get a little bit of a cough and then they're fine. Whereas northern elephant seals, at least some of them, develop that really violent pathological reaction. So this is the conservation story that we were called into. Northern elephant seals, the population has rebounded, but they seem to have this deadly so-called immunopathology because it seems to be the immune system that's causing the pathology rather than the parasite itself. So we've been investigating why. This is Francis Gulland, the head veterinarian at the Marine Mammal Center at the time, the person who first alerted us to this interesting and important problem. Here's Stephen Gorin, who was a postdoc with me and Bridget Van Holt here. At the time we started this work, he now is on the faculty at Berkeley. He works a lot at the Marine Mammal Center, and this is a live, but not too well, seal in the seal barrow, which they use to push patients around in the clinic. This work was also received important support from, again, Alec Downey, graduate student in my lab at the time, and Bridget Van Holt, dear colleague in EEB. So again, this syndrome is not observed in other harbor seals, or other phocids with whom the elephant seals share this environmental exposure risk, this infected fish risk. So we're really trying to figure out why. This is very much ongoing work in progress, so I don't have many answers for you, but I want to give you a sense of what we're doing and what we're finding so far. So we find, first of all, that lots of elephant seals seem to be exposed and only a small subset are getting sick. So now we're trying to understand why those some seem to get extremely sick. And we're interested in this both at sort of the proximate mechanistic level. You know, why do their neutrophils seem to escalate out of control? What's going on in the swarm of neutrophils in their bodies? But also evolutionarily, why are those few so seemingly susceptible to this immunopathology when they encounter the worm? We're doing cell culture methods. This is a photo of Alec and members of Roxanne Beltran at UC Santa Cruz's lab sampling blood from an anesthetized elephant seal at Año Nuevo State Park. We can culture their immune cells in media alone or in the presence of microbial stimuli, and then we are able to study immune gene changes in immune gene expression. And so far we're finding really huge immune dysregulation in seals that are exhibiting signs of the lungworm infection. We're also doing work on the underlying genetics, doing a genome-wide association study with cases and controls, so the worm positive and worm negative. We did initially a sort of a low density sequencing approach, and we have now just completed whole genome sequencing. But the lower quality sequencing so far shows a huge genetic susceptibility factor, where there are a few genes at which that basically are explaining entirely this susceptibility to the immunopathology. So our work is very much not done. That's why I'm not able to show you very much of it. But what we're wondering is whether this history of being hunted to such a low bottleneck may have left them with a few bum genes. You know, maybe this is the result of genetic drift following that bottlenecking event. But we also are bearing in mind that northern elephant seals are pretty exceptional mammals in other ways. And so just a few examples here. They are at sea over 7 months of the year. They dive 2 kilometers deep quite routinely, multiple times a day. When they're diving, their heart almost stops, their lungs collapse, you know, so little oxygen, so cold. I mean, and the immune system we expect to be involved in mitigating all of these things, both the thermogenesis, the repair of damage, et cetera. So we're bearing in mind that we may be studying quite a complex evolutionary history here, not just genetic drift, but perhaps former adaptation to their extreme lifestyles. So ongoing research, just to give you a sense of what we're finding, our data thus far suggest that these pathology-associated genotypes are in about 10 to 20% of the seal population right now. It's not 100%, which is good news for conservation, but it's still a pretty good fraction. And we suspect there's ongoing selection due to lungworm exposure. What's the flu going to do? I don't know. So an example of the kind of proximate level question we're trying to address, do neutrophils of those really acutely susceptible seals have especially sensitive receptors, or are they just making more cytokines? Like, what are the genes involved in conferring that susceptibility? What are the mechanisms that lead to the immunopathology? And at the evolutionary level, as I also sort of already sort of indicated, we're interested in dissecting the roles of genetic drift as well as prior or current adaptive value in some of the traits they need to lead their extreme lives. And for that latter question, we're undertaking some comparative analysis across marine mammals who also dive pretty deep. We're going to find out whether there are immune signatures of adaptation to those extreme environments. So final slide. The mammalian immune system, I think you can see from the neutrophil example, even that one alone, is quite a powerful and yet can be a perilous system. It's a very rewarding kind of system to study. With the rewilded mice, we put the lab mice outdoors and find huge changes to their immune phenotype, and we're in the process of quantifying the contributions of environment to those phenotypes. And along the way, I think we are improving what's called the model mammal. It's supposed to be the mammal that represents us all for a lot of biomedical research. And I think by giving them some neutrophils to work with, we're helping in that pursuit. And then last but not least, the wild northern elephant seals. This is our conservation case for studying genetic and environmental contributions to immunity. Just dissecting the causes of this disease, I think, is interesting and important, but also I hope we can inform prospects for conserving the species. So with that, thank you for your attention. I look forward to your questions.
Okay, well, thank you. And so now we're at the discussion part of the afternoon. And for those here, we have a couple of mics that will be running. And for those of you online, please send in your questions and we will receive them over here and I'll pass them on. I see you, but we would like the first question if at all possible, to be from a student, a postdoc, somebody early in their career, and I see right there. Thank you. So not much of a break after all.
That's all right.
Thank you for the talk. Going back to the rewilded mice piece of the talk, during this season, so when you release them for 1 to 2 months, and then you bring them back and then you do your measurements, how does it compare with the life cycle of the neutrophils that you showed from how much of like a season, what percentage of the total life cycle does it represent in your
yes,
judgment?
thank you for that. Yes, one thing I did not reveal about neutrophils in this talk is that they're among the shortest lived cells of our immune system. So they turn over really quite rapidly. In fact, once they've swarmed, they often just blow themselves up and leave a big mess. In fact, neutrophils cause pus. While we're in my favorite zone of talking about gross things after lunch. So neutrophils have a really short lifespan, and that's probably why I would emphasize to you that the production of them is so ramped up by the time outdoors, right? It's the individual cells are definitely changed and activated by exposure to fungi in the environment, but the whole system ramps up production because of that fast turnover. You wouldn't get more neutrophils in the blood if you didn't ramp up production.
That was brilliant. For the elephant seals, speculate about if you just left the system where it is, would the very susceptible to the inflammation genotypes just get washed out of the population, do you think?
Yes, thank you for that. I would say that let's leave aside the flu for a minute. If the primary immune selective agent is lungworm, then yes, I think we would expect the frequencies would just decline and decline over time. One thing that I didn't mention is Stephen Gorin is an expert in studying ancient DNA, and he's actually been able to go back in time to museum specimens and even archaeological specimens of the northern elephant seal to look at the allele frequencies of the frequencies of the alleles conferring this acute susceptibility to immunopathology, and they went up markedly around the time of the bottleneck, and then they've just been waning ever since. So it really does look like ongoing selection weeding those out.
So we have a question there, and then after right there.
In connection with the elephant seals, obviously you're dealing with a believed to be extinct population in which 100 individuals were found, and obviously resultant lack of genetic diversity in that group. What insights has your study shown for recovery of other isolated populations of otherwise believed to be extinct species, that is, remnant small populations? Do you have some insights as to that and to the vulnerability of the resultant populations growing from a otherwise believed to be extinct species?
Yeah, thank you for that. Very important. So I'm channeling Stephen now because he's much more of an expert in this than I am, Stephen Gorin. I think he would say that there's a lot of context dependency. It depends on quite how long they were at a small population size, what their lifespan is, all of that. But I think overall he feels that the elephant seals are a bit of a message of hope because despite that small bottleneck size, they have maintained quite a lot of immunogenetic diversity, like for example at the major histocompatibility complex, the MHC locus, which is where conservation geneticists often look to see about how much heterozygosity remains in a population. The northern elephant seals have retained quite a lot of that. They are rather inbred though, as you said. The inbreeding per se doesn't seem to predict susceptibility to this infection, but it clearly has other ramifications. But overall, Stephen feels that they are in remarkably good genetic shape, better than you might expect. So he's developing methods to find out what other species that have gone through this kind of bottleneck might also exhibit signs of, for hope, for optimism.
Hi, Dr. Graham. Thank you so much for this. I just want to know, regarding the mouse experiment, how long do these types of mouse live, like relative to the 2 months they spend outside? Because we were talking about how the cumulative life experience alters the immune response. So I just want to know something about that.
Yes, thank you. Another important point. The longest lived mus musculus probably live on the order of 2 to 3 years, but the average one lives a few weeks, right? So it's super heterogeneous. I would say that we're studying them from early young adulthood into sort of middle age. We're capturing, and because we're not studying predation, we're prolonging their lives, right? We're capturing some of that middle chunk of life, but definitely not capturing the whole lifespan. One thing we would really like to do is undertake breeding outside so that we have some pups born out there who are experiencing the real world from day one. We expect the impacts on their immune systems to be even larger than the effects on the adults that we've studied so far. But there's, as you might imagine, some logistical constraints and such that come into play that have kept us from studying the whole lifespan so far.
Danny?
Thank you.
Has a question.
Thank you. So really interesting. The case of marine mammals, so they came back into the ocean, over the Cenozoic. Their skin, various aspects of mammals would kind of be challenged by this marine environment with regard to infection. Skin would be a big one. Lungs, as you mentioned, would be a big one.
Yep.
Does this sort of enter into the immune systems of these pinnipeds and of other marine mammals? I mean, or if you look at the composition of the immune system of marine mammals versus non-marine mammals, is there something distinct?
That is fantastic. That is a burning question for us that we don't know the answer to yet, but we are trying to systematically study it by studying terrestrial sister taxa of marine mammals for each of the independent returns to the sea to ask whether there are consistent immunological changes required by return to the sea. I would expect yes, because cytokines of the immune system, including those that are required to fight off worm infections, are essential for thermogenesis. Adipose tissue, fat tissue, interacts very strongly with the immune system. In fact, in most mammals, fat tissue can become lymphoid tissue, and so I wouldn't be surprised if the blubber and that sort of thing was important immunologically in multiple ways. So I expect the answer will be yes, but I don't know. I'll have to keep you posted. We're working on it.
So I'm going to interject with a question of, so what are they doing at 2 kilometers? And do you see any differences, are they being tracked to the depths that they go or the environments that they're in at 2 kilometers for the ones that are with and without the infection?
So Roxanne Beltran, the behavioral ecologist at UC Santa Cruz, is tracking them all the time. She has them with little GPS systems on their backs, and she can track how far they are out at sea and how deep they're diving and all that sort of thing. What they're doing at 2 kilometers deep is looking for food while avoiding great white sharks. And the females are better at it than the males. The males take more risks and end up, especially when they're juveniles, they end up being eaten by sharks along the continental shelf, whereas the females go out further, dive deeper, seem to get away from the sharks. Your question, though, about what that has to do with this infection, we don't yet know. We would expect that the females should be more exposed than the males because they're foraging deeper and further out. But that's the opposite of what we actually see in the clinic. We see more males getting very sick. So I don't think it's an exposure difference between the sexes or among individuals, but we don't know for sure yet.
There's a question there. Over here.
Sort of based on that, is it possible that that difference in observed female and male is that the females are out farther and less likely to be just stranded, so they're less likely to show up in those, or is it or is that already controlled for, so you know it's more present in males?
Yeah, that's a very, very good point. I have to say that compared to laboratory mice, it's really, 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 really hard to observe these animals, right? But therefore, we always have to have that caveat that you have just raised, that they're really hard to observe. But when we're looking at the pups and the yearlings, the females and males are both present near shore. They have to figure out how to fish and all that kind of stuff. And so the capture rates, or the rates at which they show up at the Marine Mammal Center, are comparable. It's just that the females are more often there for propeller injuries and other things that they risk being out in the sea where humans are at work, whereas the males are the ones with this infection. So we think it is a real sex difference in exposure, but with the caveat that you raised, for sure.
Yeah. There's a question up here. Oh, let's wait for the mic because we have people far away.
While the mic is coming, while they're diving, they take a nap.
Good for them.
I forgot to say that. It's very important. They corkscrew down taking a nap until they get, isn't that cool? All right. I'm always up for hearing about naps in nature.
Well, thank you for the presentation. I thought it was very accessible, especially for someone who doesn't come from an ecology or biology background. I just want to confirm whether I understood something right. So is the explanation for the northern elephant seals' sensitive immunity because of the mass hunting that they experienced?
Yes, so I apologize for not being clear. Part of the problem is we don't know yet. The work is very much underway. But one of our hypotheses is that being hunted down to the bottleneck may have left them with some genetic variants that have had to be weeded out ever since, basically, because they're not great, right? But we don't know that for sure, and I'll have to, again, ask you to tune in a year or two, and I hope I have a better answer.
I would love to hear again.
Okay, thank you.
There is a question there and over here.
Hi, Andrea. Thank you for that. I had a question about the rewilded mice. I know you said that they don't exhibit any of these 30 rodent pathogens, but do they exhibit any other sort of pathologies being out there in the wild?
Yeah, we were very worried. We've even released some genetically knocked out gene mice and everything. We thought, ooh, good luck, guys. But they seem to be really, really fine. We have almost no cases of concern about their health. They get a ton more exercise, of course, than the lab controls that stay in the little cages. And so if anything, I'd say it's the opposite. I mean, we have struggled to measure things like their muscle tone and all that kind of thing, and we haven't quite figured out the right mouse scale way to do that. But as far as we can tell, they're not only fine, they're actually in better physical fitness. Yeah.
We had a question here. Andrea?
Thank you for a great talk. Going back to the elephant seals, when I think of the elephant seals, I think of, the bulls fighting. Do you think that there are trade-offs between, having that immune response to heal from those battles and this lungworm infection?
Yeah, absolutely. That's another one that keeps us up at night. We're wondering much the same. We find that from yearling stage, when they are not yet different in body size, right? Eventually there's about a 4 or 5-fold difference in body size, huge sexual dimorphism. But when they're quite young, the body size dimorphism is not apparent, but they're immunologically already very different. And this is work led by Alec Downey. We're still in the process of quite figuring out what we've learned. But we have done the cell culture, quantified the immune responsiveness, and it would be really interesting then to compare that to the pathological, because those are healthy animals, right? Sexual dimorphism in the healthy animals, how do the immune pathways identified as being dominant in the males map on to the ones associated with the pathology? We can't wait to answer that question. But
Thank you.
Yes, this is the joy of talking about work very much in progress at a talk, right? I don't have very good answers to most of your questions.
There's a question over here.
Hi, I had a question about the rewilded mice and thinking about potential reasons or mechanistically why fungi as opposed to another type of microbe might be eliciting this kind of response with regards to neutrophils, and if there's any kind of known comparison maybe within the marine environment or different environment of something that is upregulating neutrophils in this way.
Yeah, thank you for that. So first of all, compared to other microbes, the difference in fungal diversity and abundance in the field versus the lab is huge, because lab mice, despite the fact that they're in these very controlled environments, they do have some bacterial symbionts, and they have very few fungal ones, and so the effect size for the fungi they acquire is just huge. So I think that's part of what's going on. That's part of why fungi got shown in red. It's a much more marked difference between lab and field in this system. But on the other hand, we were not surprised at the fungus-neutrophil connection, because in those clean mice in the clean box, if you give them fungi, they will make lots of neutrophils, right? So the ability of fungi to induce neutrophils is not surprising. I think what is surprising is that in just a week or so, we can completely remodel the whole neutrophil production compartment. But thank you for that.
You might have already answered this and I missed it, but once, if you give them the fungi and it triggers more neutrophils, does that persist? So for whatever the life of a mouse is?
This is another thing that keeps us up at night. We've been trying for quite a few years now to undertake what we call de-wilding, right? We rewilded, we see these big effects. How much of them would persist if we brought them back into the lab? And for a long time, the veterinarians would scream at us if we proposed bringing these animals back in the lab, right? Because they're trying to keep all the other mice clean. But just last year at the NIH, we actually were able to have our first de-wilding experiment. It wasn't a wild success, long story about NIH vivariums, but we hypothesize, and the data so far seem to bear it out, that immunological effects persist even if the microbiota returns to indoor standards.
And where's that?
Right? Because the immune system, a lot of what it does is, of course, and this is something else I didn't talk about, but it retains memory, right? And so the immune memory compartment is really stimulated by time outdoors, and we expect that would persist even when de-wilded.
Brian. So while the mic is coming, where does that memory reside? I mean, I can't imagine these cells live very long themselves, right?
Well, that's right. Some of the cells live longer than you might think, but the cell lineages can live for a really long time. If you have a cell that contains your memory of the flu you got in 2009, as long as it divides periodically, that memory can persist in your bones. Yeah, yeah.
I guess it's too early to say, but it'll be fascinating to find out for the seals if the flu is worse in the hyper responders or the others, or that's the basis of a trade-off possibly.
Exactly, yes. We are, it's not the highest priority for the vets, but they are getting some samples for us to do the genetic testing. We can think of reasons why the susceptibility alleles for the lungworm would be the same as susceptibility alleles for the flu, but then we can also think of other hypotheses too, and we just don't know which way it's going to pan out. If it's the same susceptibility alleles as for the lungworm, it might be a smaller outbreak.
But it might just be specific alleles.
It might. Yeah, the southern elephant seals, like almost 70,000 of them were killed by the bird flu 3 years ago. So we're hoping the northern elephant seals don't have the same fate.
All right, folks, I got the signal from the bosses back there. So we're going to have to thank Andrea again.
Thank you all for your questions.
And I believe you'll be not vanishing teleporting, but there could be some other questions of you.
Correct.
Exactly.
Always happy.
No teleporting machines yet. Now, before you leave, I have 2 announcements for one additional presentation on Wednesday, March the 17th, St. Patrick's Day, I believe. It says Wednesday, but it means Tuesday.
Tricked you.
So Tuesday, March the 17th, which is St. Patrick's Day, at 4:30 p.m., Professor Catherine Hayhoe, who is the chief scientist of the Nature Conservancy and also a professor of political science at Texas Tech University, will be giving the Taplin Lecture, which is entitled Finding Hope in a Warming World. And I really hope that you join us for the presentation and the discussion. I think it should be very, very exciting. And then on Tuesday, April the 7th at 12:15 p.m., again at the same time, we will have our 3rd HMEI faculty seminar of the semester. And it'll be Professor Ian Borg, Associate Professor of Civil and Environmental Engineering and at the High Meadows Environmental Institute, who will be discussing partitioning of PFASs and other organic contaminants between water, air, and minerals. So looking forward to seeing you there, and enjoy the rest of the afternoon.
Thank you. Thank you, Blake.